We have start. Wow. So many.
Okay.
Okay. Hello, everybody. Now the program has a pleasure to has a Ciência Meio-Dia, né, international in English. Okay, Dr. Lukasz, is an honor for us. Né, you presentation here for us, your job, your work, and I ask Deborah if you introduce Dr. Lukasz. She knows better than us. Okay, thank you, Deborah. Thank you, Dr. Lukasz.
Thank you, Dr. Andre. First of all, I would like to thank to Dr. Lukasz because he accepted our invitation and are very preeminent in science and give an excellent lecture about major essential nutrients. And he's very, how can I say, an excellent science and very, very close person. So it's very nice to have this relationship with Dr. Lukasz. Dr. Lukasz is of University of Alabama, United States, and I will ask to him to present his a brief of his career. That is a long career, and talking about his experience. And again, thank you, Dr. Lukasz.
Awesome. Thank you so much for the very nice introduction and giving me the opportunity to share some work that we have been doing here in my lab. What I'm going to do, I am going to share my screen. So please let me know if you can see it.
Yeah, we can.
Okay. Let me just get the potent options so we can easily I can just, use it. Okay, so again, thank you for the very nice introduction and giving me the opportunity to share some of the work that we have been doing. You probably are aware of the topic of today's lecture. I will be focusing on a class of dietary molecules that are kind of overlooked usually, and I would like to try to give answer a question whether these molecules that we ingest with our food do they have the potential to be actually another class of essential nutrients that we still do not recognize as essential nutrients, or maybe they are just overstudied class of molecules that really do not have that much of value. Before I actually go into the details, I would like to always like to thank my entire team, and this is some of the part of the team, and it's very difficult to have all the people in the same picture, like especially when everyone is busy. And I also would like to thank. So obviously, what I will be sharing with you, it's a collaborative work between a lot of different lab members throughout the years. And it would not be possible if it was not for the funding agencies that are willing to spend some money to let us do this work. So I'm going to start with something that probably kind of sounds like a cliche, because everyone knows that healthy diet is good for you, right? Everyone knows that. It's something like, it's not a big of a, discovery to say that. In this particular slide, you can see that there are certain dietary or health habits that are associated with health. And in this particular case, it is actually associated with decreasing the risk of developing certain neurodegenerative diseases, such as Alzheimer's disease. So you see that exercise and certain dietary patterns, especially like Mediterranean diet, is rich in plant-based food, but also grass-fed, grass-finished meat, and also seafood. Those types of lifestyle choices have been associated with lowering the risk of developing Alzheimer's disease. And as you can see, I would like to pay you to pay attention to certain things here, because one of these things I will be discussing in detail. So in terms of the healthy brain, there are several hallmarks of that are kind of associated with lowering the risk of developing neurodegenerative diseases, namely lowering the inflammation. And this is something that I will be specifically focusing today. So those types of lifestyle changes have been associated with lowering the inflammation, but also they have been associated with increasing insulin sensitivity and also increasing the level of what is called BDNF. BDNF is a so-called brain-derived neurotropic factor, a molecule whose higher concentration has been associated in the brain with lowering the risk of developing Alzheimer's disease and other neurodegenerative ailments. As you can see, that the type of diet here, which we can consider as a Western type of diet, has been associated with actually increasing the risk of developing different neurodegenerative diseases, including Alzheimer's disease. So as I said, this is some kind of a cliche. We know that healthy diet is actually good for you, but the question is, do we really understand what's in the diet that really makes it healthy? And this paper that actually kind of came out 3 years ago really resonated with me. This is the paper that you can see the citation on the bottom of the slide. So basically, as of right now, when we think about diet, we majorly focus on macromolecules. So, the quality of protein, fats, and carbohydrates, and vitamins and minerals. But so it adds up to 150 different key nutritional components. But it has been estimated that with our diet, we ingest as many as at least 26,000 other small molecules. And we do not really fully understand what those molecules actually do upon ingestion. Sometimes actually people refer to those molecules as nutritional dark matter, because we know that it's there, but do we really know what it does? We really don't. And actually, not so long time ago, actually, there was a paper here in Nature Medicine urging scientists and also healthcare practitioners to pay more attention to using food as medicine. And on the previous slide, I actually showed you that there's actually a new field that is actually being developed. In bigger schools, they actually have already the departments of nutritional psychiatry, where they are looking how diet and lifestyle choices affect actually a lot of different neurological disorders. And if you would look at this particular paper, you'll see that they do have some recommendations, one of them being that a dietary prescription should also be on the table, not only the prescribing the medicines, but also we should be paying more attention to the food choices patients actually are having. So when we think about diet, I mentioned that there are like at least, as of right now, we kind of focus on around 150 different molecules, but and one of them are actually vitamins. So you can see here like examples of those vitamins that I'm pretty sure that all of you are familiar with. But actually, I'm quite curious if you have ever actually heard about vitamin P. This is actually something that many people have never heard about. And this is a term that actually it's not like it's not my group that coined it. It was actually introduced by this gentleman, Dr. Szent György, who is the Hungarian biochemist who discovered vitamin C, and he received Nobel Prize for the discovery of vitamin C. But while he was working on his vitamin C, he was isolating vitamin C from vegetables such as paprika or lemons. And while he was actually isolating these vitamin C, he was also co-isolating a group of molecules that phytochemists will instantly recognize those molecules as flavonoids. And he called those molecules vitamin P. In all those different models that he was using at that time, he actually noticed that the animals were actually doing much better when they were co-administered vitamin C and what he called vitamin P. But back then in the 1930s and 40s, they were not able to actually prove the vitamin effects of the flavonoids. So the concept of vitamin P was actually discarded. And the other reason I'm showing you the structure is this, because this is going to play a pretty important role further down in the presentation, where I will be trying to draw your attention to a specific element of this structure, which I am actually circling here, alpha-beta unsaturated carbonyl, which every biochemist knows is a quite reactive chemical moiety, and might be, in our opinion, might be responsible for the biological effects of these molecules. But that's what's going to happen further down the road. So the question is, why do you, we want to, I want to tell you why we actually really started focusing on this particular group of those at least 26,000 different molecules, because as I said, there are so many of them. Why would we pay particular attention to this class of molecules? So very recently, there was a lot of papers that actually showed a strong, and these were the epidemiological data, a strong correlation between a higher intake of those particular group of molecules and lowering the risk of aging-associated diseases, specifically neurodegenerative ailments. So these are the press releases. Those are the papers that actually, some of the papers. There was actually another one in May this year. So if you would go to Neurology, you will find them. This is like a flagship journal of the American Academy of Neurology. And all those high-profile papers were actually strongly pointing to the fact that this particular class of molecules is strongly associated with lowering the risk of aging-associated diseases, and specifically Parkinson's and Alzheimer's. So in general, neurodegenerative ailments. But obviously, we're not the first group that have ever looked at those class of molecules. If you have ever looked, if you're going to look into the literature, you'll see there are hundreds of, or even thousands of hundreds of thousands of papers that focus on flavonoids. Here you have actually examples of different molecules that are currently actually being tested even in clinical trials. You see the number of Scopus citations about those particular class of molecules, a number of registered clinical trials. And you will recognize certain molecules that actually belong to the class of flavonoids like genistein, epicatechin, lutein, quercetin. Those are actually specifically flavonoids. But I actually challenge you to actually, with the wealth of all these different trials and papers, to identify even a single molecule that has been actually registered as a drug. None of them was actually registered as a drug. Zero. So much research, no. Even a single molecule being registered as a drug. So the reason actually it happened like this is when medicinal chemists look at the flavonoids or other molecules that have this particular structure that I showed you, the alpha-beta unsaturated carbonyl, which makes it inherently reactive, they instantly actually classify it as a pan-assay interference compounds, PAINs. It means that those molecules give positive effects and or readouts in a lot of different assays that are being used to assess the biological activity of these molecules. They also have other, medicinal chemists have actually a lot of other, insults. They love coming up with all those different insults towards these molecules. The other one is IMPs, which they call them improbable metabolic panaceas. So molecules that are good for everything, which means they are good for nothing, right? But if you actually go deep into all these different studies, most of these molecules have been tested mostly in vitro assays, where you isolate, for example, a single protein and you expose it to this inherently reactive molecule flavonoid. And we think that that's part of the problem. So it is actually important to the fact that a molecule reacts with, for example, an isolated enzyme in an in vitro tube. It does not mean that this exactly same reaction will happen in a context of a living cell or a living organism, where there are a lot of other factors that are involved. So at this point, we have this discrepancy that kind of led us to pursue this project. On one side, we have all these high-profile studies showing a strong association between the higher intake of flavonoids and lowering the risk of Alzheimer or other neurodegenerative diseases. And on the other hand, we have all those different claims saying that those molecules are useless drugs. So what we are thinking is that these molecules will never be able to meet the criteria of what we consider to be a drug, which by, for example, FDA standards, it's a molecule that it's inherently specific in interacting with one specific target. So with all that knowledge, we kind of started with a hypothesis. What if this class of molecules might be a understudied class, or poorly understudied class of potentially essential nutrients? So when we approached this problem, we definitely wanted to start with testing these molecules in a context of a living organism. We didn't want to test it in vitro assays. Obviously, there are those of you who have studied flavonoids, they know that there are many of these molecules, hundreds of these molecules. So when choosing an animal model, it is important to pick one that or two that will be actually, if you want to test all of the different molecules, that will be actually, it will be feasible. So obviously it is not possible to do it right away in a mammalian models. So we decided to start with actually simple, to test those molecules in the context of a living organism using invertebrate models. And we have used the toxin-induced Drosophila model of Parkinson's disease, as well as C. elegans model of Alzheimer's disease. What I will show you further will be only data focusing on this particular model, but what I really want you to bear in mind is that every single molecule that we found to be effective in this particular model, it actually was also affecting in this model. And what we found to be ineffective in Drosophila model was exactly ineffective also in the C. elegans model. So focusing on this Drosophila model of Parkinson's disease, as I said, this is a toxin-induced model. So in this particular model, we actually induce Parkinsonian symptoms in Drosophila using paraquat. You might have heard about paraquat. Paraquat is a herbicide that actually has been associated with actually development of Parkinson's disease, including humans. And it actually also causes Parkinsonian effects in flies. It actually decreases their survival, decreases their mobility. I'll show you the data. It also kills dopaminergic neurons in their brain. I'll show it to you on later slides. So what we did in this particular project, we actually, before the flies were exposed to this Parkinsonian causing effect paraquat molecule, they were either pre-fed with a flavonoid. So we first gave them flavonoid and then hit them with paraquat, or we also tested another approach where we basically co-administered both molecules at the same time. Here you have examples of two different molecules that are flavonoids, and I'll later explain why I'm particularly focusing on these two molecules, but it actually applied to any other flavonoid that we tested in the same way. After, so in both pre-treatment and the co-treatment. First, we tested the survival. So we checked how the treatment actually affects the survival of the flies. Whenever you're going to see a green bar, it will be actually showing you the data for gardening A. If it's going to be black, it's going to be only control. Sucrose, blue, it's always going to be only paraquat. So you see that the treatment with gardening A, so this flavonoid, actually increases the survival of the flies. They survive better if you treat them with paraquat. It actually also improves their mobility that is tested in something that is called climbing index. In flies, similar similarly as in humans, flies actually, their mobility is impacted in when they are exposed to paraquat. But as you can see, gardenin A actually rescues these effects. Interestingly, a molecule that has a very similar structure, gardenin B, you can, which whenever you're going to be seeing a orange bar, it's going to be corresponding to gardenin B. As you can see, it does not, gardenin B, contrary to gardenin A, does not actually improve survival. It does not improve the mobility either. So it's quite interesting. So the reason I'm actually showing you those two molecules is that after the hundreds of different molecules that we have tested, actually gardenin A turned out to be the most neuroprotective. And I will show you some of the data later on, because we already completed a mouse study with this molecule. That's why I'm just kind of single out these two molecules. But we tested a lot of other molecules, and you see some of the data here. I am not going to be going over the details because it's not really that much relevant. But what I really wanted to show you and to tell you is that there was one particular class of molecules that kind of stood out. So if you would, the molecules that actually did not have any substitution here at this C3 position were the one that were actually the most active. As soon as we introduced something here like hydroxyl group in 3-flavonols, the molecules were not really effective in our model. So after identifying this class of molecules that were neuroprotective, we wanted to actually investigate what might be the molecular mechanism of action. We started to dive deep into the what's happening in the Drosophila brain. So what you are seeing here are actually isolated Drosophila brains, and those yellow, green spots are actually GFP tagged dopaminergic neurons. In case of Drosophila, those dopaminergic neurons actually cluster together. They are actually present in several different clusters, and you can actually easily count those neurons under the microscope and check how a specific treatment actually affects it. And it was actually interesting, and we can actually count it. So you will see the differences between the treatment of sucrose paraquat, gardenin A, and gardenin B. I hope you can easily see that, for example, paraquat treatment significantly decreases the number of those green spots, meaning that it's actually decreasing the number of dopaminergic neurons. You can score those neurons under the microscope, and here you have the quantitative analysis of those dopaminergic neurons in two specific clusters, the PPM and PPL1 cluster, and hopefully you can easily see that paraquat treatment decreases the number of dopaminergic neurons in those two clusters, while gardenin A, but not gardenin B, have a protective effect. You see that the numbers of those dopaminergic neurons upon treatment was actually higher compared to just paraquat treatment, meaning, okay, these molecules seem to be protective towards this dopaminergic neurons. So we wanted, okay, we see that dopaminergic neurons are being saved with those molecules. How do they do it? Obviously, one of the very first things that we would look into is something that people keep looking for, quite a while in terms of natural products is, do they have an antioxidant effect? You probably have heard, like, those molecules are antioxidants. So yes, we did look into it. One of the very first things that we look into, we look at the levels of malondialdehyde. Malondialdehyde is a product of oxidation of phospholipids. So the more oxidative stress you have, the higher the level of malondialdehyde, meaning that there is a higher oxidative stress happening in the organism. So you can see that paraquat actually significantly increases the levels of oxidative stress measured by the levels of malondialdehyde. But interestingly, both gardenin A and gardenin B decrease the levels of malondialdehyde. We also looked at the levels of the expression of CNC. CNC is a Drosophila ortholog of NRF2. You probably might have heard of NRF2. NRF2 is a master regulator of oxidative stress, including humans. So it makes sense that actually paraquat increases the level of expression of CNC, or homolog of NRF2, because flies are experiencing actual oxidative stress upon paraquat treatment. But what was interesting, we also saw a further increase in the level of expression of this particular gene when the flies were actually treated with those molecules, both guanine A and B. We also looked at some of the genes downstream to that CNC, because CNC or NERV2 is a transcription factor, so it regulates the expression of a lot of different genes. So what we have seen is actually the same pattern. The treatment with actually those molecules further increased the levels of expression of some downstream genes. And you remember, guanine A, but not guanine B, was neuroprotective, but both seem to have antioxidant effect. So it kind of led us to a conclusion that the antioxidant effect is definitely not the solely responsible for the neuroprotective effects that might be associated with the guanine A activity. And as I said, we tested multiple different molecules and we have seen exactly the same patterns. But what is actually quite interesting, and I wanted to kind of draw your attention to it, that if we actually exposed flies to only flavonoids and we did not treat them with paraquat, we did not see a significant increase of the CNC expression. So this actually further increasing the expression of CNC was only observed upon the challenge, like oxidative stress, which is quite interesting. So we started looking into other things, and you might have heard about inflammation, specifically neuroinflammation. And I'm not sure if anyone has ever heard about a term called inflamma-aging, which basically means that while we age, and this is true for humans and also flies, the level of chronic sterile inflammation increases, and it has been associated with a lot of aging-associated diseases, including neurodegeneration. So we look into the neuroinflammatory responses. One of the things that we have looked into is the levels of expression of relish. Relish is a transcription factor that is in human ortholog is an NF-kappa B, which you probably have heard about. It's also a master regulator of innate immunity response. And again, we have interestingly seen that actually paraquat increases neuroinflammation by which was indicated by increasing the levels of expression of relish, and guanine A, but not guanine B, actually decreases the levels of expression of this gene. When we used relish null flies, so a knockout line, we actually lost the neuroprotective effects of guanine A, which would suggest that actually this neuroprotective effect is definitely driven through the relish IMD pathway, at least in flies. We also looked at other markers of neuroinflammation, such as the levels of nitric oxide. High levels of nitric oxide have been associated with neuroinflammation. And as you can see, paraquat increases the levels of nitric oxide, and guanine A actually brings it down comparably to a known inhibitor, but guanine B does not. We also looked at the level of expression of an enzyme, nitric oxide synthase, that is involved in the synthesis of nitric oxide. And again, paraquat increases the level of expression of this protein, and the guanine A, but the guanine B does not do it. And guanine A actually decreases the levels of expression of nitric oxide synthase. We actually look at the levels of expression of relish for other neuroprotective molecules, and we've seen that all basically neuroprotective flavonoids actually brought the neuroinflammation down. We also look at other markers. We look at markers of, for example, apoptosis or programmed cell death, and you can see that actually paraquat increases the level of activity of caspase 3, which is one of the main caspases involved in the process of apoptosis, so the programmed cell death. And guanine A, similarly to an inhibitor, actually inhibits the activity of caspase 3, preventing cells from undergoing the apoptosis. We also look at the levels of expression of some pro-apoptotic genes like Reaper. And again, paraquat increases the level of expression of pro-apoptotic genes, but guanine A and not guanine B actually decreases the level of expression of those pro-apoptotic genes. So long story short, we have seen that all of these different molecules actually have a lot of different types of activity. So they are not typically molecules that would be designed, considered as drugs, because they have a lot of different targets that they might be affecting. Similarly to vitamins, because vitamins, if you would like to, registered as drugs, you would also hit the same wall. They are usually quite pleiotropic in their mechanism. But if there is one home take message from this, is that I would like you to remember that every single molecule that we have tested in terms of their neuroprotective effects, what really kind of stood out was the fact that they were able to actually modulate the neuroinflammatory responses. So that's another part of the talk that I'm going to be focusing on the next, let's say, 15 minutes. But before I go into exploring further the neuroinflammatory effects and how those molecules actually affected, I want to kind of follow up on what I already told you, is that why we kind of single out gardenin A from all those different molecules that we tested in Drosophila. Gardenin A actually was the most effective molecule that we actually compared to any other neuroprotective flavonoids. So we decided to move forward with these molecules and test it in a mammalian model of Parkinson's disease. And it's a genetic model. It's the model that those mice are genetically engineered to actually express the alpha-synuclein. And as you can probably know, in one of the hallmarks of Parkinson's disease in humans is the accumulation of misfolded and hyperphosphorylated alpha-synuclein. And that's what happens actually in this mice. So we used this model where we treated the mice with 2 different doses, 25 and 100 milligrams per kilogram. We treated them for 3 weeks, and on week 2 we started the behavioral test. We actually tested them for the memory using the conditioned fear response. And open field. We also tested their mobility in a digitate assays. After those tests and after completion of this study, the mice were sacrificed. We isolated the brains. We looked at the levels of expression of certain genes, similar to those that we looked in Drosophila. So again, antioxidant response and neuroinflammation. We looked also at histopathology, looking at the levels of accumulation of alpha-synuclein. And I'm going to go quite quickly through those slides so that I can focus on neuroinflammation. But if you have any other questions, I will be more than happy to answer those questions after the presentation. So long story short, gardenin A in mice actually increased the memory, improved the memory in the mice. It actually also improved their mobility compared to control. As you can see, it also, so you have the black bars here as the 100, the gray here is 25. It increased the levels of expression of certain synaptic proteins, so it actually saved the synapses. It had anti-inflammatory effects as measured. We look at the level of TNF-alpha, interleukin 6, which are pro-inflammatory, and it actually increased the expression of certain antioxidant genes. So you can see the pattern is exactly the same as we have observed in flies. What else was quite interesting, we also saw that the treatment with gardenin decreases the level of phosphorylated synuclein. So the higher the level of synuclein, the higher the neuropathology. And we have actually also saw the increasing levels, although it was not statistically significant, levels of tyrosine hydroxylase, which is basically an enzyme involved in the synthesis of dopamine. And, so specifically looking at the health of dopaminergic neurons. So if there's something that I want you to remember from this part is that gardenin A tested in a mammalian model of Parkinson's disease actually was neuroprotective and actually hit exactly the same pathways, at least the ones that we have tested. We have also also tested the presence of gardenin A in the brain, and we were able to actually prove the presence of gardenin A in the brain. So we were proving that it actually makes it to the brain. But I want to focus the last 10 minutes or so on the neuroinflammation story, because this is something that we have been exploring further. So I told you that every single molecule that we have tested, and it was also proved in a mammalian model, seemed to have regulated neuroinflammation. And as I said, neuroinflammation has been associated with a progression and pathology of numerous diseases, including Alzheimer's and Parkinson's. So for a very long period of time, it was believed that inflammation is a self-resolving process. What it means that once inflammation starts, there are pro-inflammatory molecules that are secreted that start and maintain the inflammation. Then the organism takes care of these molecules and inflammation ceases. But now we know that actually inflammation is a actively resolved process. So there is another class of molecules, small molecules, that is actively involved in the process of resolution. So you have a separate class of molecules that drive inflammation, and there is a separate class of molecules that is actually involved in the process of resolution of inflammation. Here you have the names of these molecules: lipoxins, resolvins, marazines. And if you look at the chemical nature of these molecules, these are the derivatives of essential fatty acids, such as here you have here I'm actually specifically showing a pro-resolving molecule in flies, because we have been testing it in flies. But flies also cannot synthesize the essential omega-3s, so they have to acquire them through the diet. So we started looking, so this molecule that I'm actually showing you here is considered to be a pro-resolving, so anti-inflammatory molecule in flies. So when we started looking at these molecules, we instantly noticed that all the different molecules that we have tested that have actually this anti-inflammatory effect have something in common. And the commonality was actually the fact that these molecules are actually soft electrophiles. All of them have this alpha-beta unsaturated carbonyl group. And remember, the fact that these molecules have this group is associated by the medicinal chemists with the fact that they should be excluded from any serious drug discovery endeavors. Why? Because this makes those molecules inherently reactive. There is a possibility that these molecules will react with a lot of proteins or other nucleophiles in the cell, which will lead to side effects. So there is this fear. These molecules are inherently reactive. They are not good candidates to be drugs. But we actually ingest a lot of these different molecules with our food, and so are vegetarians more likely to be, having all those different side effects associated with the fact that those molecules are inherently reactive? So we started to dive deeper into the possibility of reactions between those soft electrophiles and some proteins in the context of a living cell. So for a long period, so what the medicinal chemists are really fearing is the non-enzymatic post-translational modifications. So a reaction between a reactive electrophile like this molecule with some electrophile responsive proteins, because the molecules that we have known to date that are the result of such reactions are usually associated with different types of diseases. A good example of such a molecule is a hemoglobin A1C, which is known to be measured as a marker of the control of type 2, for example, diabetes. And if you look at this type of a molecule, it's actually a molecule non-enzymatically change hemoglobin through a reaction with glucose. It's a glycated, not glycosylated, but glycated hemoglobin, where the more glucose you have, the longer it actually interacts with hemoglobin, the higher the likelihood that you will have a higher concentration of this changed protein. Another class of those proteins that are changed with sugars are, you might have heard about, advanced glycation end products, which are kind of hot topic right now, and they're also associated with different diseases. So the reason the medicinal chemists are fearing of these reactions is that for a very long period of time, we've only been associating those non-enzymatic post-translational modifications with oxidative or metabolic stress and disease. But we are trying to, and there are other groups that are also trying to raise this question, can those reactions also happen in a context of a healthy cell? Because if we accept the fact that life evolved from a random chemical reactions, we should also accept the notion that those types of non-enzymatic reaction may still occur in highly evolved systems, such as eukaryotic cells. And I always, when I'm talking to medicinal chemists, I always show them this particular example. This is a small molecule here, dimethyl fumarate. This is a molecule that is currently, for example, approved by FDA for the treatment of SM. And in Germany, it's actually, I think, approved to treat psoriasis. But if you would look up the molecular mechanism of action of this molecule, a lot of data point to the fact that this molecule can actually potentially non-enzymatically, post-translationally modify certain nucleophilic centers in proteins, but not any type of proteins, only those that actually have a specific type of amino acids associated with that nucleophilic group. So the non-enzymatic post-translational modification reactions that we have been fearing that are because there are no enzymes that regulate them, there might be actually a lot of other factors in a context of a living cell that allow the control how these reactions happen. A good example is, for example, the pH of the cellular compartments. Why would pH matter? Well, as you probably remember from biochemistry, the protonation or deprotonation of this nucleophilic center strongly depends on the pH, and only deprotonated cysteines or any other thiols are actually strong nucleophiles, so they're more reactive than a protonated one. The accessibility of the nucleophilic center matters. The interactome, how the specific protein interacts with other things, matters. So we started looking at the possibility that some of those dietary molecules can possibly non-enzymatically post-translationally modify certain regulatory proteins in our cells. So we started with pretty simple reactions where we exposed a simple nucleophiles to some of our molecules that we have tested without the presence of any types of enzymes. And yes, we were able to actually prove that those adducts between the nucleophile and electrophile actually happen. Obviously, this was in vitro, but so we were not satisfied. So we started to moving forward towards looking at how those reactions actually happen in a context of proteins in the cell. And first, we started looking at two specific types of proteins that we knew before might be involved in the neuroprotective effects of these molecules. So NRF2, so CNC in Drosophila, and NF-kappa B. And interestingly, if you would look at NRF2 in the cytoplasm, it is actually associated with KEAP1 protein that has a lot of nucleophilic centers. So upon modification of those nucleophilic centers, this complex, actually, the KEAP1 dissociates from NRF2, allowing its nuclear translocation. And it's actually very similar to NF-kappa B. NF-kappa B actually has a lot of nucleophilic centers. So we started looking at the possibility that those transcription factors that have so many nucleophilic center can potentially respond to those electrophilic dietary molecules. So here you have an example of KEAP1, the primary sequence. And how we study it, we basically feed the flies, looking at the if those and then trying to search for those non-enzymatically post-translationally modified proteins in those flies. We do it using MALDI, so the isolated protein is then digested using trypsin, and we look at the peptide. So the way you would see if a specific peptide is modified is that you should see an additional mass corresponding to the mass of this electrophile that should have been added to the specific peptide. And yes, we were able to see it, for example, for certain KEAP1 and certain electrophiles. And this is an ongoing project. One of the things that I wanted to point out is that we started to wonder is like what happens with those non-enzymatic post-translational modified proteins. These are proteins where you have a covalently attached something. So and there's not to be any enzymes that are involved in the process of regulating those proteins. So how the cell would actually regulate this how would deal with this particular modified proteins. One of the things that we have been considering is that the possibility that those types of proteins might be removed through a lysosome autophagy pathway. And interestingly, we have seen that in this the treatment with actually those electrophiles such as gardanine A increases the levels of expression of autophagy genes. We have been also looking, one of my students is also looking at the possibility that what happens if to the level of physiological pro-resolving molecules in the presence of these electrophiles. And interestingly, we have also found that certain electrophiles like cardenine A, thymoquinine, or even aspirin, they also increase the levels of physiological pro-resolving molecules. So that might be another way those molecules actually provide neuroprotection. With all of that, I actually would like you to, if there's one thing that I want you to remember, is that we think that those molecules that have been disqualified as potential drugs, because they are so biologically active, we think that they may constitute a class of what we call of potentially essential nutrients, that and we call them potentially essential dietary soft electrophiles that might be involved in the process of resolution of inflammation. And those classes are not only flavonoids, by any other lipophilic membrane associating molecules that have the ability to either, that form or are molecules with this alpha beta unsaturated carbonyl group. And with that, I would like to thank you for your kind attention, your kind introduction, and I would be more than happy to answer any concerns and any questions that you may have.
Oh, thank you for your presentation.
Thank you.
Yeah, it's good. It's nice job. Amazing job. Anyone has a question for Dr. Lukasz? Alguém tem alguma pergunta, professor Lukasz? Alguma questão, alguma coisa que a gente...
Eu tenho.
Você, Débora.
I should have studied some Portuguese.
Okay. Dr. Lukasz, I found the lecture brilliant.
Thank you.
And I have a question.
Sure.
Is there already detail in the literature about how these essential nutrients would act on epigenetics and modulation of gene expression? And how could this influence the emergence of diseases or their prevention?
Yeah, so there is actually, so obviously I mentioned that I think in my talk that we are not obviously the first group that has ever kind of approached the studying of these class of molecules, and there have been a lot of people who have been looking at. Many groups have been focusing majorly on, for example, antioxidant effects, but now there's actually a lot of people that are starting to pay attention on the effects of, how these molecules may affect epigenetics. For example, the modification of certain classes of deacetylases or the process of methylation. And there have been some data that actually show that these molecules might be actually affecting gene expression by actually affecting epigenetics. The question that we are trying to answer is like, is there any, can these effects, including, the epigenome regulation, can be associated with the fact that these molecules can actually directly interact with certain proteins? So, for example, would it be an interaction with, let's say, sirtuins that are the deacetylases, right? So I would assume that those things that you are asking might be downstreamed through what we are actually investigating. But yes, there are a lot of there is some data that point to the fact that these molecules actually strongly affect gene expression by regulating actually epigenome. I hope this answers your question.
Yes, and the most important thing is that we only think to epigenetics as a cause, but could be the consequence.
Yeah.
And the alteration, these proteins are the cause. So it's very interesting.
Yeah. So I think it's I think that when we approach this problem is like if if anyone has ever actually studied phytochemicals, people have, especially, you know, in the realm of drug discovery, people have very strong opinions about it. So because there's so much data in the literature, and people kind of consider this class of molecules to be overstudied, and it basically there's nothing interesting to find there anymore. People looked at it, they are just useless. But I think that the problem is that what really tarnished the reputation of these molecules is the fact that many of these studies actually come from in vitro assays, where, you know, and as I hope actually I was able to convince you that these molecules are actually inherently reactive. So they would react in a test tube very well with whatever you expose them to. However, this reactivity is actually modified in the context of a living cell. So those reactions between the electrophiles and nucleophiles are not as random as we thought. They are actually very context dependent. So it is actually very important for us to try to figure out how those reactions may be happening in a context of an intact cell, because the fact that something reacts in a test tube does not necessarily translate to what's going to happen in a context of a living organism. And, you know, there is so much data showing that these molecules are actually good for you. And why would, you know, so what? There's probably something that, you know, they are definitely doing something, but it's not as easy to you know, decipher it. So that's why hopefully our studies will at least shed some more light into the, you know, or give people some other ideas where to look for.
Deborah? Another question, Deborah? No?
No.
Anyone? No. I have
Our
some.
students are shy.
The student here is ashamed. Okay, I will try to to ask it because it's now my main job. I work with cancer, but guardenina, I don't know the accent. Guardenine, guardenine A appear the good molecule. Okay, okay. Guardenina is present in what species of plants, what vegetables, concentration, guardenine?
Yeah, so guardenine A actually is, interestingly, a molecule that is, so it's a polymethoxyflavonoid. So you saw that it has a lot of methoxy groups, and it's actually, interestingly, it's the major molecule, the major plant that it was identified from. It's called gardenia. So most of you might have heard about it in context of just a plant that is basically, you know, has a beautiful flowers, and you would see it in the garden. So normally it would not be a typical thing that you would eat, but what I want to actually tell you is that very similar molecules actually are present in plants from the very same family. It's a Rutaceae family. So citrus, for example, they have a lot of polymethoxyflavonoids. They have nobiletine, tangeratin, sinenzetine, hesperitine. They are all polymethoxyflavonoids, and those are the molecules that we also have found to be neuroprotective. But actually, it's actually quite interesting that you mentioned cancer, because there is actually, there have been a lot of interesting work that I've seen recently in a context of, for example, NRF2, so the master regulator of antioxidant response in a diet. So it has been shown that actually diet, when you are a cancer patient and undergoing chemo, having a diet that is actually high in those molecules activating NRF2 may be actually counterproductive, because it actually upregulates the NRF2 response in cancer cells as well, and actually allows the cancer cells to survive chemotherapy. So this is another very, caveat of this work is like, we obviously know the diet impacts how we respond to treatment, but this is something that people have not been paying that much attention. So for example, you might be, if someone is a cancer patient and undergoing treatment, and a lot of these patients are also taking, for example, dietary supplements because they want to help themselves to, okay, I want to do everything possible to kind of, fight this cancer. But there's some data that point to the fact that you might be actually doing yourself harm by actually, getting some of the more molecules that might be, helping cancer cells actually survive the treatment with the chemotherapeutic, which is quite interesting. So, yeah. But the gardening by itself, getting, circling back to your question, it is not as far as I know, it is not present in any edible plant. Maybe I'm wrong, but it's mostly in gardenia. But there are a lot of molecules very structurally similar in, for example, oranges or any other such citruses.
Okay. And about another signaling pathway, NF-kappa-B. Okay, we work with NF-kappa-B. A guanine A inhibits activation of NF-kappa-B because of decreased inflammation. Okay.
Yeah, and you know that inflammation and activation of kappa-B has been associated with cancerogenesis. So yeah, so we've seen what we have actually seen is that it actually all the molecules that we have seen to be neuroprotective seem to be actually affecting the inflammatory responses. So one of the things that we have looked at was actually the regulation of the level of expression of NF-kappa-B. And yes, we've seen that gardenin A and any other molecule that we have found to be neuroprotective, they have been actually decreasing the levels of expression of NF-kappa-B, and then the levels of nuclear translocation as well, because obviously that's what matters.
Okay, I'm curious about the gardening A. You isolate it or you buy commercially to?
So I think gardening A is commercially available. So we mostly actually for what we have done, we actually bought the purified one. But so you can actually get it from I think there are several vendors that actually sell it. We are actually right now wrapping up our mouse paper. So like hopefully by the end of this month, we will release the preprint, and then we will have also and submit the manuscript. But we definitely will share it with the scientific community in a form of a preprint. But yeah, gardening A is available, I think, commercially, because that's what we kind of done in the past, where we actually got it from a vendor.
Well, thank you for your answer, for your time, for presentation, the excellent presentation.
Thank you so much, and yeah, I hope if you happen to have any questions, I actually am going to go ahead and share my screen once again, so you will see that at the end. Can you see it now?
Yeah.
Yeah. So you can see my email. Can you know? Yes. And also my Twitter handles for the lab and my personal Twitter. So like if you feel that you have some questions or comments, or you want to just link, just please shoot me an email, and I would be more than happy to connect.
Thank you.
Thank you, Deborah. I'll see you.
Thank you, Dr. Lukasz. Yes, you were brilliant. Very nice. Very nice.
So much. Thank you so much, and have a wonderful day. And again, thank you so much. And I hope that the students also enjoyed it.
Yeah,
they love
absolutely.
it.
Yeah.
Love it.
Thank
you very much.
Bye.
Bye.
Thank you very much, Professor. It was a pleasure.
Thank you. It was a pleasure to meeting you all. Thank you.
Thank you very much.
Bye.